All right, good morning. Let's start the review. Just gonna basically go through some of the key concepts that you should know for this upcoming quiz. And I know we did a lot of discussion on the experiment and stuff like that, but you wouldn't have to know like the details for all of them. Okay, so just kind of know the broad concepts, right? So one of the experiments that we talked about was Hershey and Chase. Hershey and Chase. And they worked with these bacteriophage, which are viruses that infects bacteria. And from their experiment, the key finding, the key conclusion was that DNA and not protein is the hereditary material. Right? And they basically use radioactive sulfur and phosphorus to label the proteins or DNA component of the bacteriophage. Okay, so that's basically what they did. Now, we also spent a lot of time talking about the Mendelssohn and Stahl experiment. Okay, so again, in that experiment, they were working with E. coli, and they were grown in heavy nitrogen, which was then transferred to regular nitrogen to grow, right? So initially, what happened was after several generations growing in the heavy nitrogen, all of their DNA is going to be consist of heavy nitrogen. Okay, so I forgot what color I use in class, but like, you know, red here is heavy DNA, okay, because it was grown in heavy nitrogen 15, right? Then it was transferred to a medium that contains regular DNA, and then, you know, we would call that like a lighter DNA. Right? So like if DNA that consists of just the regular nitrogen, and that's the light DNA. Okay, so this is N14, and then the other one was N15. Right? And then you can have something that are mixed, right, in between. Okay, so whether it's semi-conservative like that, or whether it's like, you know, dispersive like that, right? The idea is you would have DNA that is mixed of heavy and light DNA. So, you know, we can call that like intermediate, right? Intermediate DNA, right? A combination of 15 and 14. Intermediate as in like the weight being intermediate. All right, and so the E. coli, after being transferred to the lighter medium, it was allowed to grow for one generation, and then they extract the DNA and spin it down, and then they do it again for the second generation. So what you must know is, you know, how it would look differently if DNA replication follows various models. Like what if it follows the conservative model? Okay, conservative model, if you extract DNA after one generation, you would have a heavy band, which represent the original DNA. And then the entire new copied would be using light DNA. So you would expect to have two bands like that. And then if you go ahead and do another round of division, the original DNA will still be there. And then everything else that are being copied would now be lighter DNA, nitrogen 14. Okay, so if those were the results, then they could conclude it is conservative. If it's semi-conservative, which by the way is the correct model, you would expect to see the DNA being in the middle, right? Because semi-conservative means you will have one strand that's completely new, and then the other one is completely original, right? And if you go ahead and do it for one more round, you would have the mixed band in the middle, just like before, but now we will have an additional lighter band, which represent brand new DNA. Finally, there is the dispersive model. And for that case, you will have just a mixed band in the middle, right? Because dispersive means it will always look like that, right? It's mixed together, not restricted to one side of the DNA ladder. It's just like all over the place, right? So these are the expected result that you would get if DNA follows these various models. And of course, these are not the correct ones. Their experiment shows that it is the semi-conservative model that was the correct one. Okay, hope that makes sense to you. And I'm just gonna go through this one with you. This is of course DNA replication. I'm just gonna go through some of the enzymes with you again, just so you have a like a review on them. So this one over here, this one is the gyrase. Okay, so the function of the gyrase is to relieves tension in DNA, right, as it is being unzipped. And what enzyme would be responsible for unzipping the DNA? That's right, the helicase, right? And that would be represented by number 2 right here. And there we have our helicase. Okay, so it unwinds DNA, and also it creates the origin of replication, right? Remember, in prokaryotic cells, there would be a single origin of replication. Like if this is the original DNA, the helicase would create a bubble, right? That's an origin of replication. For eukaryotic cells like us, right? We have a lot more DNA, so it's necessary to create multiple origin of replication. Okay, so that's like one difference between the two. Now, after you separate the DNA, they have a tendency to come back together, which is why we're gonna need to have these little balls here, right? These little balls, number 3, those are the single strand binding protein, right? Single strand binding protein, and their job is to keeps the strand, DNA strand, apart. You don't want them to come back together. You want to give access to other enzymes, right, to the nucleotides. Okay, so that's the main ones. Now, there's also this concept about like leading strand and lagging strand, right? And that's all because of the enzyme that is responsible for building the new strand, and that would be this guy right here. So that, of course, is DNA polymerase 3, right? And I think we have a couple other ones here. That is another DNA polymerase 3. And then there is another one here. Okay. So of course they can only replicate in the direction of 5 prime to 3 prime on the new strand, and they cannot initiate the process, right? They cannot start the process, which is why we are going to need to have these primer, right? So there's going to be an RNA primer here. There's going to be another RNA primer here, here, and here. Okay, so the RNA primer, which I should label RNA primer, is going to be laid down there by this enzyme, right? This is square enzyme. So that would be the RNA primase, right? It positions, it helps position the DNA polymerase 3 basically by laying down a few RNA nucleotide. Okay. So once the primer is put in place, then the DNA polymerase 3 is going to carry on, right? And it will build a new strand, right? It's going to go in the direction of 5 prime to 3 prime on the new strand. Right? So here we go, another 5 prime to 3 prime, 5 prime to 3 prime, like so. Okay. Because DNA strands are anti-parallel to each other, so one strand will be continuous. Okay, so this strand is the leading strand. Okay, it's continuous, like as in like it's being created continuously, and it goes towards or into the fork, the replication fork. Whereas the lagging strand, right, they are synthesized discontinuously, and it contains these little fragments, right? You probably remember the name, the Okazaki fragments. And also it is going to be moving away from the fork. Okay? It moves away from the fork. Any questions so far? Okay. All right. So at the end, right, like when the DNA polymerase 3 approaches the primer, right? Remember, this is actually going to fall off, right? Fall off and replaced by DNA polymerase 1, right? Which will remove the primer. Okay, and ultimately we're gonna need to seal the gaps. We have to join the Okazaki fragments, which is what this guy is for. And that is the ligase. Oh, that's a terrible color. Ligase, L-I-G-A-S-E. Okay, that's to connect the Okazaki fragments, right? Now we talk about the fact that if this is the end of a DNA, for example, right, then there is nothing coming from upstream to replace the primer, right? That's why every time you do DNA replication, you're gonna end up losing a little chunk at the end of your DNA, right? And to prevent the loss of critical DNA information, we need to have telomeres, right? So like at the end of your DNA, of your chromosome here, for example, you will have the telomeres at the end, okay? So they are non-coding repeats of TTA, GGG, and their function is to protects the end, protects chromosome ends. Okay, and it's important to know that they get shortened after each DNA replication. When it's critically short, right? Critically short, then not allow to divide. Right? The cell would become senescent. There's going to be growth arrest. You cannot allow the cell to divide again, because if you divide another round, which means you're going to have to copy your DNA one more time, you will start eating into the genes that are located at the end of the chromosomes, right? So that is not good. What's the enzyme that re-lengthens the telomeres? Anybody remember? Yep, germ cells and stem cells have telomerase to reset telomere length. Okay, so that when we pass on to our next generation, they're not gonna start with like critically low DNA, right? So that is that. All right, let's see what's next here. Give me a second. I just need to know what's going on here. All right. So, what is this thing called? Anybody remember this picture? Oh man, I cannot make it smaller. What do we call something like that? Anyone? Oh, where's the chat box? Oh, there it is. What do we call this picture? I will give you a hint. It starts with a K. Ooh, finally, something that, oh, I was gonna say something Jay doesn't know, but she knows. This is the karyotype, very good. Karyotype, okay? It's an orderly arrangement of chromosomes, right? Okay, and is this a male or female? What is the sex of something of the person with a karyotype like that? Yeah, that's a male, right? Because it is XY. Okay, so you should be able to tell. If I show you a picture, yeah, there is a Y exactly. If you see XX, right, then it's gonna be female. Very good. Okay, so just a couple of terminologies to refresh your memory, because this is what what this session is about. It's about refreshing your memory. And so there is like a chromosome, right? And that's chromosome 1 from mom and chromosome 1 from dad. I mean, it does have the chromosome 1. It could be like chromosome 4 and chromosome 4, right? And so, you know what? I'm just going to call this chromosome number 5. Okay. Oh my goodness. Number 5 here. Number 5. So these are homologous chromosome. Homologous chromosome, right? Okay. And what happens is during DNA replication, you will end up having the identical copy, right? And they're going to be stuck together. They're going to be stuck together like that. Okay. So this is number 5, also number 5, number 5. Okay. But this is still considered as one chromosome, right? And then here is another chromosome. Okay. Now these are identical. This one and this one, they are exactly the same in terms of like genetic material. And what do we call them? They are stuck together. Do you know what we call them? Right. Those are the sister chromatids. That's right, sister chromatids. Okay. This and this are also sister chromatids. Okay. And they're connected at the centromere. Centromere. All right.
Sir,
Yeah.
before you move on, is there like a big difference between the chromatids and the chromatins?
Oh, yeah. They are different. I know they sound the same, right? Okay, so chromatin, TIN, is just DNA wrapped around proteins. Okay, so that's like the normal form of DNA that exists within the nucleus, right? And then before cell division, they condense more, and then they become these chromosomes. Okay. If you remember in class, there's a picture, right, like of these DNA, and then it wraps around these little balls, right, like strings around a yo-yo, right? So that's like the chromatin, TIN, and then the chromatid, TID, that's referring to when after you replicate that they're stuck together. Okay. So homologous chromosomes, sister chromatids, yeah, basically that's that. Okay, so let's quickly talk about the fact that there are 46 chromosomes, right, in human, for all the somatic cells, bodily cells, anything that's not a sperm and egg, 46 chromosomes. And then there would be 23, nope, there will be 22 autosomes. Okay, autosomes are chromosomes that determines everything about you except for the sex, right? Except for the sex chromosome. So X and Y doesn't count. And then we have 2 sex chromosomes. Okay, XX or XY. Okay, these are everything else, like number 1 to number 22. They determine everything else, like, you know, hair color, eye color, blood type, earlobe, number of fingers, all that stuff. All right. Oh, and by the way, I just want to backtrack here a little bit. Sorry, I'm just like going through things as they come to my mind. And everything I tell you here are important, right? For the purpose of the quiz. You have to at least know everything that is talked about here. So for the homologous chromosome, they're same length, same size. Their centromere are going to be at the same location. And more importantly, they would have genes that are in the same order, right? But sometimes they might have a different version of a gene, right? So like here, you might have gene big B and small b, right? So these are different versions of a gene, which we call allele. Okay, allele, different version of a gene, okay? Like, brown eyes and blue eyes, that's different version of eye colors. Okay, let's just quickly go through mitosis. Very simple version of it. You have to know how to identify what phase they're in. But I'm just gonna quickly do one here. Okay, so this is at the late prophase already. Okay, so like you got these chromosomes that have already duplicated, right? That's why they have the sister chromatids, right? And I have chromosome 1 here, 1, 1, 1, 1, and then the smaller one would be chromosome 2, 2, 2, 2. Of course, like in the actual cell, you would have like all the way up to chromosome 23, right? The sex chromosomes. But for simplicity, we're just gonna do it like that. Okay, so this is gonna be prophase. I'm not gonna write everything down, but I mean you have that in notes, right? But I'm gonna verbally tell you the gist of it. Okay. So chromatin has now fully condensed into chromosome. You can see them. It's easier to move them around, right? Like think about moving to a new place. You gotta pack up your stuff, right? So it's easier to carry. So you pack up the chromosome, condense them into this form, and you can move them around easily. Of course, you have your centrioles, which are now at the opposite end opposite ends of the cell, and they will be sending out these spindle fibers, right, which will help guide the movements of the chromosome. Of course, the nuclear envelope has now disappeared, and then all these things are out. Remember, I'm not going to ask you to describe the mitosis, but you should know, things that are going on in each phase. So how many chromosomes do we have in this picture? Hopefully your answer is 4, right? 1, 2, 3, 4. If they're stuck together, we count them as 1, right? There's no reason why. It's just the way it is. Okay, don't get too hung up on it. After the prophase is the metaphase. In metaphase, they all line up in the middle, right? They all line up in the middle, like that. And of course, you have the spindle fibers here, doing like a tug of war and try to put them in the middle. And these things are the centrioles, right? Okay, so how many chromosomes here? Still 4, right? They are still stuck together. And then we're gonna have the next phase, which is the anaphase. And in anaphase, we will separate the sister chromatids. They get pulled apart. Right? They get pulled apart by the spindle fibers, and they will get dragged to the opposite poles of the cell. Now, how many chromosomes do we have in this anaphase? Okay. Anyone else want to participate? Okay, let's do a poll, huh? Let's do a poll. Launch the poll. If you think it's 4, please choose A. If your thing is 8, please choose B. If your thing is 16, please choose C, and then D is 2. Okay. Thank you for participating. Well, the good news is nobody chose 16 and 2, so you know that I'm happy. But half of you chose 4, half of you chose 8. The correct answer is 8. Okay, 1, 2, 3, 4, 5, 6, 7, 8. Okay, if they're not stuck together, you have to count them separately. Okay? If they're not stuck together, you have to count them separately. So the answer is 8. Okay, please remember that. And then finally, telophase, you're gonna have the little snowman, right? There you go. And then the chromosomes will be at the individual cells. Okay, so there you go. Okay, so this is gonna divide soon, right? And the telophase, sorry, telophase, and the division of the cytoplasm is called cytokinesis. All right, everything is in your notes. Any question at all before we move on? I think that's a pretty good spot to stop for number 6, for lecture 6. Now we should move on to number 7. Okay, number 7. Oh, by the way, of course, I forgot to talk about this very important thing. There is the cell cycle, right? Cell cycle, which is like mostly is going to be interphase, and this is the actual mitosis, right? And don't forget, there's going to be like G1 here, checkpoint. There's going to be the S phase, right? S phase, which is the part that you actually copy your DNA. And then afterwards, you're going to have like G1 and G2. So G1, just briefly, checks for the quality of the DNA and the state of the cell, make sure it's healthy enough to copy its DNA. And once it's copied, you want to check for the quality again, right? Did you make any mistakes or you had to fix anything, right? So this cell cycle is regulated by, you know, a bunch of green lights and red lights. Okay, there's no yellow lights, just green and red. Okay, like that. So green light means they could go ahead and divide. Red light means stop, right? And so the green lights, these are growth promoters, right? Growth promoter, right? Which are usually proteins, and they're encoded by proto-oncogene. Remember that? Right, proto-oncogene. This cytokinesis business is in my way. Let me get that out. There we go. Okay, so proto-oncogene is a normal gene that is responsible for promoting cell growth. And then these ones, they are growth inhibitor. Okay, growth inhibitor. Okay, which are encoded by tumor suppressor gene. Right, tumor suppressor gene. And now, tumor suppressor gene also plays a role in DNA damage repairing, right? Anything that kind of fix things up would be a tumor suppressor gene. Okay. So for this test, that's all you have to know about proto-oncogene and tumor suppressor gene. Okay, I'll test you more on them in the final test, but for now, that's enough. Okay. So if the growth promoter, if the proto-oncogene becomes an oncogene and the tumor suppressor gene loses its function, loss of function mutation, then the cell cycle is going to go out of control, right? It will be always on, so it will keep on going through the mitosis, and it would have nothing to stop them, right? And that creates a neoplasm, which is a tumor, okay, a tumor. Remember, tumors are not automatically cancerous, right? They could be benign, or they could be cancerous, malignant. Okay. Did I spell benign wrong? I don't know. Can't really spell. Oh yeah, I got it right. Okay, so benign and malignant, right? So, how do you know what's benign or what's malignant? Well, there are some characteristics, right? Characteristics of cancer that you should definitely know. Okay, so they're all in the PowerPoints, but I'm just going to list them here for you. Self-sufficient growth signal. Okay, they don't need green lights anymore. They have their own green lights. Okay, and they follow their own rule. They could divide on their own without signal stimulating them. Okay. They would ignore signals that stop or slow division. I'm paraphrasing, okay, from what I have in the notes. Okay, so not only do they have their own green lights, they won't stop, even if you try to tell them to stop. Okay, it's like my kids. Okay, stop hitting your sister. Okay, they don't care. They just keep on doing it. Okay, so they ignore stop signal. Next one is they have sustained. Again, these are in no particular order. I just write them as it comes to my mind. Sustained angiogenesis, which means they can create their own blood vessels, right? And it supplies nutrients to it. It sucks the nutrients out of your body to supply the growth of the tumor. They evade apoptosis. They won't die. They have limitless replicative potential. Keep on dividing, no problem, because they have telomerase, which makes sure that their telomeres don't get short. And last one, they have the ability to invade and spread. Okay, invade and metastasize. So these are the characteristics that are shared by all cancerous cells. Okay. Now there's that. So that's it for lecture 6, basically. That's everything that you should know. Now, for lecture 7, the main thing that you have to know is basically what we have been doing this whole week. Okay, like you have to know how to go from template strand to RNA and then to protein, and then from, if I give you coding strand, you have to know how to do that as well. You also have to know the anti-codon and then the actual codon and stuff like that. I mean, I don't want to spend too much time on that because we have been doing it all week, right? We spent a whole class doing it yesterday, right? So maybe I'm just gonna do like one example with you. Okay, so let's say we have the template strand, template DNA, let's say it is UAC, and then TAC, TGC, ACG, and then whatever, like AAA, and then ATT. Okay. So if I give you that, then what you need to do is, what do you need to do? You need to find the mRNA. Okay, and Amber already has the whole thing. Very good. So this is template, right? If I give you template and I ask you for mRNA, you have to do complementary rule. So that will be AUG, ACG, UGC, UUU, UAA. And then we have to look up the amino acid. Before that, actually, you will also be asked to find the anti-codon. Okay, that 5-mark question, you also have to know the anti-codon. And yesterday, you know, with the stuff that I made, stick it to the whiteboard and show you the relationship between the anti-codon and the mRNA. You should know that they are complementary. So this would be UAC, UGC, AAA, and AUU, right? Complementary. And then you would have the amino acid, right? So you just got to be careful, like on the test, right? You look up the amino acid based on the mRNA, right? So this is going to be MET. And I told you I will be giving you the table. Okay, just do this slowly. In fact, you know, this question is going to be on the last page. Do that first, get it out of the way, right? So you're not going to get confused later. And then stop. That's it. Okay, you're able to do something like this, you get 5 marks. So good. Yeah, I mean that's basically it. I don't think there is anything else. I mean, there is the stuff about like promoters and terminator, but then we reviewed that on Tuesday, beginning of Tuesday's class already. So I don't want to go through it again. A couple of other stuff that you should maybe know. Let's say I have 50 amino acids in the protein. Okay, how many nucleotides do we need to code for that? What's the minimum number of nucleotide that is required to code for something like this? Okay, you know what? Let's make it 60, and then I'm gonna do a multiple choice. Like, how many nucleotides do we need to code that? Is it 20? Is it 60? Is it 30? Is it 180? What's the minimum number of nucleotides that are needed to code for a protein that has 60 amino acids? Okay. Well, thank you for those who have chose something. So let's take a look at here, right? Right. That's a 1, 2, 3, 3, 6, 9, 12, right? Right. So look at like that. That's 12 nucleotide, and the 12 nucleotide codes for 1, 2, 3, 4 amino acid, right? Each amino acid requires 3 nucleotides to code for it, right? So if it's 60 amino acid that we need, then we need 60 times 3, right? So we need at least 180 nucleotides to go for it. Okay, assuming that there are no like introns and stuff like that, right? That's why I said minimum, right? Because you know there might be more, and that's okay, but that's how you get the answer. Okay, so that's it for the review. Of course, you're welcome to email me if you need to have extra help. But this is the gist of it, okay? As far as the content on the quiz go. All right. Well, thank you for coming on this Friday early morning. You enjoy the rest of the day. Okay. Have a good one, guys. Bye for now.